Okay, this is chapter 13. This is the microbe-human interactions. We're going to start talking about the whole disease process now and how microbes and humans interact to either cause disease in human tissues or how the human system fights back. This is going to be over this whole process of infection and infectious diseases is going to tie into our next chapters of immunology, and those are going to be chapters 14 and 15. So this unit is going to cover chapters 13, 14, and 15. The human host is the primary area of contact with microbes in the environment. You have contact and infection and disease that kind of is this vicious cycle, this continuum where bacteria make contact with the human host, they infect or get inside the human host and begin to attach themselves or get down into deeper tissues, and then they're going to get to a certain concentration where you begin to have symptoms and you have the disease process. Body surfaces are constantly exposed to microbes. If you think about the air that you're walking through every day, you have trillions of microbes. Your clothes, when you put them on, your clothes are not sterile, so whatever microbes are on your clothes then are going to be on your body. Inevitably, at some point, this exposure is going to lead to an infection. If you have infection with a pathogenic microorganism, these organisms are going to figure out a way to penetrate the host defenses and enter into the tissues where they can use those tissues as sources of nutrient to allow for multiplication. The pathologic state is going to result when you have some sort of infection that causes damage to tissues, and these tissues then or organs will then be in a disease state, and you begin to see symptomology. Now, when we talk about infection in the disease state, the disease can be something as minor as a small infection like a pimple or something major like flesh-eating bacteria. It's all going to depend on the microbe and the tissues that get damaged and the degree of damage that takes place. When we're talking about an infectious disease, however, this is going to be the disruption of tissues or organs that are caused by microbes or their products. A lot of bacteria produce proteins called toxins, and these toxins will get into tissues and begin to destroy those tissues, and you end up then with an infectious process, an infectious disease. When those toxins get in there, some of the microbes, or even all of the microbes, the offending organisms, are going to be gone, but the toxins are left to bind to the tissues, and it's the toxins that are causing the disease rather than the microbe itself. We all have our own resident flora, our own resident biota, and this is basically bacteria that use humans as a habitat. If you read in your chapter at all, one of the things that they talked about is that cell for cell, microbes outnumber human body cells 10 to 1. That is huge. If you sit there and think about, we have trillions of cells. Let's say we have 1 trillion cells. If our microbiota is going to outnumber us 10 to 1, that means we have 10 trillion bacteria on and in us at any given time. This is an enormous number when you sit there and think about the number of bacteria that are part of your normal resident biota. Our normal bacteria, we've talked about this a little bit in lab, about we have our normal flora, and that normal flora can also have, we can and divide that into our resident versus our transient flora. We're going to talk mainly about our resident flora, and this is going to be bacteria that has a stable, non-pathogenic relationship with the human host. This is our microbial armor. If we don't have this microbial armor, this is kind of like a big open wound, and bacteria can just get in, and we will be at the mercy of the organism. But our normal biota is going to have often an antagonistic effect to these pathogenic organisms, and we'll talk about what that antagonistic effect is going to be for bacteria here in a little bit. We acquire our resident biota by the places that we are, where we go, where we live, the people we associate with. The body provides a very wide range of habitats. We've got wet habitats, we've got dry habitats. We've got places on the body where it's warmer than in other places on the body. So we have a wide range of microbes as well. The microbes that are in the mouth are not going to be anything like the microbes that are on the skin or in the gastrointestinal tract or the urogenital tract. Because these areas are different with regard to their environmental makeup, the microbes that infect those are going to be significantly different. Most of the areas of the body that come in contact with the outside environment are going to be those things that we're going to call our portals of entry. We have the vast majority of our microbes are going to come in contact with the body, then they're going to be removed or destroyed by the host immune system, and these are going to be like our transient flora. So these are not going to colonize like our resident flora. But you can have, because the body is so exposed, you can have pathogenic organisms that light on you or in you, and your host immune responses will take care of it before it ever has a chance to grab a foothold. This is a table that's in your textbook that shows you the sites where your normal flora is going to reside, and you can see that the skin and its mucous membranes, as well as the respiratory and gastrointestinal tracts, the outer portion of the urethra, generally the bladder itself, is going to be sterile, but the outside of the urethra, the part that is exposed, is going to be covered with bacteria. External genitalia, as well as the vaginal tract, the external canal of the ear and the external eye, the eyelids and the conjunctiva. These are all going to be exposed to bacteria and are going to be sites where we're going to find that normal flora. These are anatomical sites that tend to be sterile. There is some information out there where you have had people isolating bacteria from some places that were thought at one point to be sterile. But the book is going to talk about these particular sites here as being sterile. There isn't bacterial life growing in any of these places. Take some time to read over this table so that you can get a good idea about where these structures are. I like this table because it actually shows you on those sites where you do have bacteria, what fungi, what arthropods are common, and then some of the remarks that you can have, for instance, if we lose the fungi, the candidus, if we lose our bacteria in the urogenital tract, the fungi that is in the genital tract, like that Candidus albicans, can cause yeast infections. And so it talks a little bit about the dynamics between all of the flora that we have in our body in those sites where we do have established normal flora. Your biota is basically your bacteria. Biota just means life. Bio means life, and this can fluctuate with a number of different reasons. Your general health. Are you healthy? People that are healthy, that eat well, that get sleep, those kinds of things, they are going to tend to have a healthier biota than someone who eats a lot of fast food, has a high calorie diet that is high in empty calories. They don't get a lot of sleep. Their general overall health and well-being is not as good. Age. As we age, our flora is going to change, and we're going to see some fluctuation in our normal flora as we age. Variations in our diet. If we go from being somebody who is a meat eater to a vegetarian, or vice versa, our flora is going to change. Hygiene. Hygiene is probably the most important aspect of our normal flora that we cannot neglect for obvious reasons. If we don't have good oral hygiene or good just overall hygiene, we are going to be potential sources of outbreaks for other people in the population. Your hormones, as well as other drugs that you're taking, these can cause fluctuations and changes in your normal biota. a lot of times those bacteria that we have are going to be very beneficial to the human host. And basically, this is going to help with the prevention of overgrowth of harmful organisms. And I talked a little bit ago about microbial antagonism. And this is basically where these normal flora are going to prevent the overgrowth of harmful microorganisms by limiting the amount of space and nutrient and environmental condition that other harmful organisms might find themselves in. For instance, the fermentation of glycogen by the vaginal lactobacilli can cause the environment of the vaginal tract to become highly acidic, and this acid environment is not suitable for the Candidus albicans that is part of our normal fungal flora of the vaginal tract. And so by keeping that pH low, that Candidus stays at subclinical levels and we don't end up with yeast infections. If that pH changes, then things are going to change. But this microbial antagonism that this lactobacilli has over Candidus will help keep the vaginal flora intact and at concentrations that are subclinical. Hosts that have compromised immune systems can also be infected by their own biota, their own normal flora. Your normal flora is not going to cause a problem unless it gets someplace that it shouldn't be. And these are what are called endogenous infections. These are going to be caused by your normal flora that are already present in the body. And because they are already present, this is your own flora. And so these can be you can have an infectious relationship, but generally, if your immune system is intact, that's not going to happen. When bacteria begin the infectious process, these organisms are going to go through a series of steps for the progression of an infection. I want to talk about a little bit of terminology, if you will, so that we can be sure that we're all on the same page as to what we're talking about. When we're talking about a pathogen, this is a microbe who has a hostile relationship with the host, and it's going to result in some sort of an infection. That infection is going to lead to disease. So this is a parasitic relationship. We can divide these pathogens into two groups. We can have our true pathogens, which are capable of causing disease in a healthy person. And then we can have our opportunistic pathogens, and these are going to cause disease when the host defenses are compromised. Our opportunistic pathogens are going to be pathogens that are generally going to be part of our normal flora, and when our immune system is down, they're going to take the opportunity to cause some sort of a disease process. The type and severity of an infection is going to depend on the pathogen's pathogenicity. This is going to be the ability of the organism to cause disease in the host. It is a concept that's going to describe the organism's potential to cause harm. Okay? If it has the potential to cause a lot of harm, then you're going to have greater pathogenicity. Staph aureus is a good example. It can cause a very minor infection, or it has the potential to cause significant infection like necrotizing fasciitis or toxic shock syndrome, both of which are lethal. So Staph aureus, even though it can cause small infections, has the potential to cause a greater pathogenicity, can cause greater harm. True pathogens are also known as primary pathogens. These are going to infect individuals with a healthy, intact immune system. This is what most of us get sick from. If we get sick with the flu or a cold or some sort of disease that we can associate with a recognizable disease, these are going to be caused by our true pathogens. Opportunistic pathogens are going to cause harm when the host immune responses are down, when our defenses are compromised, and they're going to take the opportunity to make you sick when normally they wouldn't. These become established in the body, and they are not natural to the body. So these are going to be those organisms like the Candidus of the vaginal tract. These are going to be bacteria or fungi that take opportunities when they can to cause harm. This is a table. I apparently have this in here twice. But you can see some factors that will weaken host immune responses, old age or extreme youth, so age is going to be a factor. Any kind of genetic defects in immunity or acquired immunity, surgery and transplant, organic diseases like cancer or liver malfunction or diabetes, things that are not caused by bacteria. Chemotherapy or immunosuppressive drugs, these are all going to cause the immune system to be working harder or to be dampened down, and they're not going to be as good at acting as a barrier as they normally would. Physical and mental stress. When we are under physical and mental stress, our immune system kind of gets a little wiped out, and we find ourselves coming down with maybe a cold or fever blisters, things like that. And then when your body is fighting another infection someplace else, this is going to increase the susceptibility to an infection that normally in otherwise healthy people would not cause a problem. Virulence is another term, and this is basically the degree of pathogenicity. Okay, pathogenicity is the probability or the potential for the organism to cause harm. The virulence is its degree of pathogenicity, and this is going to be established by its ability to get itself into the host. That host is going to establish itself within the tissues. It's going to enter the host. It's going to attach to that host, and it's going to survive any host response that the body might throw at it. And so once it establishes itself in the host, it now is able to cause damage. And these microbes can cause damage a number of different ways. They can do this by producing toxins, or they can induce a host response that is going to cause damage to the tissues. So you have a number of different ways that damage can be caused by these organisms. Virulence factors are any characteristic or structure that the microbe has that will contribute to its virulence. Can it stick and avoid host immune responses? Can it produce a toxin? Can it invade by getting into deeper tissues? These are all things that would be like virulence factors. Different healthy individuals are going to share a wide variety of responses to the same organisms. The hosts are going to evolve or adapt, if you will, to the organism. I'm diabetic, and say you're not diabetic, and because our individual response is different to the same organism, our individual overall health is going to contribute to how we respond. So we don't all respond the same way, even to the same organism that's causing the same disease. This is the second copy of that table. I'm not really sure why I put it in here twice, so cross out one. I think they say exactly the same thing, so use one or don't use one. I don't really care, but there are two of these in here. When the microbe is in the process of causing disease, there are 3 steps that it has to take. The first one is it has to become established, and that is going to mean that we have to have some sort of portal of entry. A microbe is going to enter into body tissues through some portal of entry. This is usually going to be some sort of cutaneous or subcutaneous membrane bound structure. It's normally going to infect the same anatomical region that is going to support that biota. And so we have a number of different portals of entry on the body. The 4 general portals of entry are the skin, the gastrointestinal tract, the respiratory tract, and the urogenital tract. Some of these major pathogens have very specific portals of entry, because if we inoculate them in a place that does not have a portal of entry, then they can't establish and they can't cause disease. It's like going through the wrong portal of entry. If you were to inoculate yourself by breathing in the influenza virus, you are going to get the flu. But if the influenza virus actually comes in contact with the skin, there is no portal of entry on the skin for the influenza virus, so there won't be any infection. So this is like going to your neighbor's house instead of your house. You can't get into your neighbor's house, but you can get into your house. You have some sources of infectious agents that can cause disease to get into these portals of entry, and these are called exogenous and endogenous agents. The exogenous agents are going to originate from outside of the body. These are going to be things that are in the air, in the food that we eat, the water that we drink, any kind of outside source that can carry or transmit an agent into the body, into one of these portals of entry. Endogenous agents are going to be organisms that already exist on or in the body. This is going to be either our normal flora, or it's going to be something called a silent infection. This is going to be an infection that's caused by that normal transient flora. Let's look at some of the ways that these agents can enter into these specific portals of entry. Let's look at how they enter the skin. The skin is a common portal of entry. It's our largest organ, and so it's got the largest surface area. If you have nicks or abrasions or punctures on the skin, these are going to be open areas, no matter how small they are, for microbes to get in. If you think about when you shave your legs or you shave your face, as you pull that razor across the skin, you are leaving small abrasions that are microscopic, but they are bigger than the microbe, and so the microbe can get into those small abrasions and can cause tissue damage. They can cause some sort of an infection. Intact skin is very tough, and very few organisms can penetrate it, but there are some bacteria that can form their own pathway using digestive enzymes or bites of other animals that can cause a break in the skin and allow for the entry of these microbes into deeper tissues. The gastrointestinal tract, this is one of the portals of entry where we're going to take in something from the outside, like food or drink or other substances that go into the mouth and end up in the gastrointestinal tract. You're going to have organisms that are going to hang on to those structures, the food, the drink, the medicines, whatever we're putting in our mouth and swallowing. Many of those bacteria can survive the digestive enzymes and the pH changes from the mouth to the stomach and the stomach to the lower GI. So they can survive those changes and then get into the gastrointestinal tract, where they can end up causing some sort of tissue issue. Thanks for the little rhyme there. Tissue damage. The best known agents are going to be the enteric bacteria, those bacteria that live in the gut. These are generally going to be gram-negative rods in the genera of Salmonella, Shigella, Vibrio. Vibrio is not nearly as common as Salmonella and Shigella, but it is something that if you eat clams and oysters that you do need to worry about. These are bacteria that live in very high salt concentrations. They can live in seawater. They can live in the oceans where you get these clams and oysters. So again, you do need to be careful with those. And there are certain strains of E. coli that can act as enteric pathogens that can cause gastrointestinal disease. The respiratory tract is probably the largest portal of entry, and we get the greatest number of pathogens by breathing them in. These microbes can be transmitted from one place to another within the respiratory tract because all of the structures are connected by a continuous mucous membrane. You get an infection in the mucous membrane of the nose, and it can easily get to the ears, to the upper respiratory tract, to the lower respiratory tract, because that membrane is all connected. So when you get a cold, it's no wonder that you have pain in the ears. You can have a sore throat, because all of that can potentially be infected by a single breathing in of these respiratory agents. The size of the microbe is also going to determine where it lands once it's breathed in. The smaller the cell, the more deeply it can be inhaled. If you've got a large microbe, as you breathe in through the nose, the hair of the nose, the hairs within the nose and the ciliated escalator are going to keep those larger elements out of the parts of the lower parts of the upper respiratory tract and down into the lower respiratory tract, and they're going to be able to keep them relatively clean. But if the elements are smaller, they're going to be able to get their way past those defenses down into the deeper tissues and cause significant disease. Urogenital portals of entry are going to be the portals of entry of many of our sexually transmitted diseases. These sexually transmitted diseases account for nearly 4% of the infections worldwide. So we have a lot of sexually transmitted diseases. Now I'm going to show you a table here in a little bit that says that when we do the math, it's about 16 million cases. But if you read your book talks about 13 million cases. So I'm not sure which is incorrect, the map or the table, or if it's a typo in the book. But regardless, we have tens of millions of cases of sexually transmitted diseases every year. Microbes are going to enter into the skin or the mucus of the penis, the external genitalia, the vagina, the cervix, as well as the urethra. Some of these can penetrate even unbroken surfaces. They have enzymes that are going to go in and start to break down some of these surfaces that allows them to get into deeper tissues. Not all urogenital infections, though, are sexually transmitted diseases. Some of these can be caused by displaced organisms. Remember earlier I said we have microbial biota that is going to be okay in one part of the body, but if it gets into another part of the body, it can cause harm. An example of this is going to be some of those enteric GI bacteria getting into the urinary tract. If you have bacteria that get from the GI tract and get into the urinary tract, they're going to cause a urinary tract infection because these are bacteria that aren't there all the time. They aren't normal to that spot, and so they're going to be opportunistic, and they're going to cause disease. Here's the table I was talking about. You can see just by doing some quick math in your head that this is going to be well over 13 million established cases each year. It's roughly 17 million, so I'm not really sure where the discrepancy is as far as the book is concerned. But again, we have tens of millions of cases of STDs that come through those urogenital portals of entry. You do have some pathogens that can use the human body itself as a portal of entry, and these are going to be pathogens that can infect during pregnancy and birth. The placenta is generally a very effective barrier, keeping most microbes out of the newborn system. You do have some microbes, though, that can cross the placenta. The spirochete that causes syphilis, Treponema pallidum, can very easily cross the placenta. There are a number of viruses that can cross the placenta, and these generally do not bode well for the newborn. Neonatal syphilis has about an 80% fatality rate. So the likelihood that your baby is going to survive if they have neonatal syphilis is pretty remote. You do have some other infections that can occur perinatally. This means that they are going to be happening during the birthing process as the baby comes through a contaminated birth canal. As the baby is born, nurses and doctors have an acronym called TORCH, and these are the diseases that they are going to look for right away on further inspection of when the baby is born. Toxoplasmosis, other diseases, rubella, cytomegalovirus, and herpes simplex. So TORCH is the acronym for that. And I'm sure when you guys get into the neonatal part of your nursing program, or if you're going into the healthcare field, that you're going to learn about these different diseases. Size of inoculum can have a definite impact on the disease. The quantity of microbes that are in a dose of bacteria can determine how big an infection that you have. The quantity of microbes that are required to cause disease is called the inoculating dose. For most agents, this is going to precede. You're going to have these organisms that are going to come in, and if they have a high enough infectious dose and you've met the infectious dose, you're going to end up then with an infection once you've reached the maximum, or the minimum infecting dose. Smaller infectious doses are going to have greater virulence. The fewer number of bacteria that are required to cause disease, the easier it is for the pathogen to cause disease, the harder it is for the immune system to get it taken care of before the disease process takes place. If we look at some different diseases, the infectious dose for cholera is 1 billion cells. So it takes 1 billion cells for us to begin to see symptoms of an infection of cholera. Typhoid is about 10,000 cells. Gonorrhea, only 1,000 cells. So you can see why we have a lot of cases of gonorrhea because it doesn't take very many bacteria to infect. There's a disease called gonococcal conjunctivitis, or oh gosh, what's the other name for it? We'll talk about it later when we talk about sexually transmitted diseases. But it's basically a perinatal infection of the eyes of babies as they come through the birth canal of a mother that has gonorrhea. And if you think about what a short trip that is down that birth canal, you are going to see that there are only 1,000 organisms. So it doesn't take very many before you begin to see an infection. Tuberculosis, it only takes 10 cells. Now, tuberculosis is very slow growing, so a lot of times the body can take over and destroy the tuberculosis tissues before you have a full-blown case of tuberculosis. But again, it only takes 10 cells. And the bacterium that causes Q fever, it's a rickettsia. You only need one aerosolized cell in order to have this bacterium basically explode throughout the body, become systemic very quickly, and you end up with this disease called Q fever. Any numbers of bacteria that are below the infectious dose is not going to result in infection. This is what we call subclinical or non-symptomatic disease. So you're not going to have any kind of infection. The second thing that has to happen, you have to have the cell get into the bacteria get into the host cell. But the second thing that has to happen is they have to be able to attach to host tissues, and this is called adhesion. Adhesion is a way for microbes to gain essentially a foothold on host tissues. These are going to be ways that they can grab on and hang on so that even as host defenses are trying to pull them away and get them out of tissues, they can hang on there. A particular pathogen, however, is limited to only the cells to which it can bind. If it doesn't have a receptor, or if it doesn't have the ability to allow these microbes to hang on, those tissues are never going to be infected by particular organisms. There are a number of different mechanisms of attachment for bacteria, fembrae, surface proteins, as well as slime layers and capsules. These are all going to be ways that bacterial cells can bind to host tissues, and it makes them very difficult to remove during an immune response. Spikes, viruses use spikes to bind to receptors on the cell, and then those host cells are going to pull those viruses in the cell either through membrane fusion or through endocytosis. If you have a eukaryotic cell like a worm or a fluke or something like that, they're going to use things like hooks and suckers and barbs to hang on. If you think about a leech and how a leech will hang on to the skin and how you have to really work to pull it off. Hookworms, they've got those barbed hooks that are going to hang on to host tissues and work their way in. The last step to becoming established within host tissues is surviving host defenses. You're going to see in chapters 14 and 15 that our immune system is a pretty amazing machine, and that it begins the whole process as soon as it recognizes that there's a problem. We have 3 lines of defense that are going to work to try and keep microbes from getting a stronghold in human tissues. Cells that are not part of your normal flora are more than likely going to encounter some sort of resistance mechanism that the host is going to throw at it to keep it from infecting. Probably one of the major processes is process of phagocytosis. Now I'm just going to touch on phagocytosis a little bit here, and we'll get into the steps in chapter 14. Phagocytosis is essentially white blood cells that are going to engulf and destroy any sort of infectious agent with these enzymes and antimicrobial chemicals that it holds inside the cell. These are going to be chemicals that are going to degrade the whole agent, the whole microbe, and eventually destroy it completely and release it back into the extracellular space where it will be completely destroyed. Some microbes, though, have antiphagocytic factors that can be used by some very aggressive pathogens to avoid phagocytosis. Leukocidines, these are going to be toxic proteins that are going to basically break apart white blood cells. The white blood cells that are used to take on the host, that is part of the host immune response. Your antibodies, your T cells, your B cells, your macrophages, these are all white blood cells, and these can be destroyed by bacteria. Streptococcal and staphylococcal microorganisms are very good at producing these. You have slime layers and capsules. These make it very difficult for a phagocyte to gain purchase on these microorganisms. They can't grab onto them and engulf them, so they can kind of slip away, and it's very hard to grab them. Some examples of this are Salmonella typhi, which is very common in the gut and causes gastrointestinal infections. And then Streptococcus pneumoniae, that is very common for the production of bacterial pneumonia. Other microorganisms have the ability to survive inside the phagocytic cell. You're going to see that when in the process of phagocytosis, one of the things that the microorganism does is it produces this vacuole inside the cell. And some of these bacteria can survive inside that vacuole, and they just remain there and hide from any sort of host immune response. Mycobacterium is a microorganism, a bacteria that can survive very well inside phagocytic cells. HIV can do the very same thing. It hides very well in macrophages, and it then is not seen by the host immune response, and so it can find its way through the body into deep tissues and cause significant damage. So we've talked about the 3 steps of becoming established. Now let's talk about disease and how some of the virulence factors can contribute to the whole process of disease and the damage that it can cause with tissues. Virulence factors, remember, we talked about earlier as adaptations that the organism uses to establish itself into the host immune system or to avoid or invade the host responses. Some of the factors are going to determine the tissue that gets damaged. Depending on the type of virulence factor, we're going to see, is it a direct damage or is it indirect damage? These virulence factors can inflict either direct damage or indirect damage. When we're talking about direct damage, we're going to use something called exoenzymes, or enzymes that are going to work on the outside of the cell. And these bacteria can also produce toxins, and those toxins can have an effect. And then you have indirect damage, where these bacteria are going to cause excessive or possibly inappropriate host responses. And sometimes you can end up then, because the host is not responding correctly or it's an overstimulation, you're going to see that those are going to cause tissue damage as well. Some of the bacteria produce these extracellular enzymes, and they can break down tissues, they can elicit damage to those tissues, and some of them can even dissolve some of the defense barriers that the host can put up to help avoid these bacteria and these other microbes getting into the tissues. Some examples: mucinase. This is a digestive protein that causes the mucous membranes that coat a lot of the open areas. Every place that we have an opening to the outside is covered with a mucous membrane. This mucinase can digest those mucous membranes, those protective coatings, and these organisms then can get down into deeper tissues and establish themselves there. Keratinase, this is an enzyme that digests the keratin that is the major component of skin and hair. So if you've got some of these flesh-eating organisms, they can essentially go through intact skin without any break in the skin. They can dissolve the keratin that's going to allow them then to get through the skin and get into deeper tissues. Collagenase. Collagenase is going to digest collagen in the connective tissues, and it's going to essentially break down tissue integrity. And now these organisms can swim their way through some of these now broken down tissues. Hyaluronidase is going to digest that hyaluronic acid that you're going to find between cells. It's the cement between the cells, and it's going to break that down. And when all of that is broken down and you don't have any adhesion between cells, now it's a very easy way for you to have microbes get past those. It's kind of like not having a lock on the door or no latch, and you just kind of can swing through the door. And so now you've got these microbes, because there isn't anything holding these cells together, that can just go right through. Some microbes can use the blood and react with the components in the blood to find their way into deeper tissues. Coagulase, this is an enzyme that bacteria used to produce blood or plasma clots. It uses the host's own blood clotting cascade to build a clot around itself, and it hides now from host immune responses. These and other organisms can produce an enzyme called kinase, and kinase is going to essentially dissolve those fibrin clots. These coagulase producing organisms are also going to produce a kinase, so they can get out of those clots. But other bacteria can also produce a kinase. You're going to see that in the inflammatory response, one of the things that happens is we begin to produce blood clots. And some of these bacteria can take those inflammatory clots and lyse them and move past the inflammatory response into deeper tissues. Some bacteria produce toxins, and these are products that are going to be essentially poisonous to any type of tissue. An organism that is not the organism that produced the toxin. When we talk about toxigenicity, this is kind of like pathogenicity. This is the power to produce a toxin. Toxinosis, this is going to be any disease that is caused by toxins that are produced by bacteria. And then toxemias are going to be diseases where the toxin is spread by the blood from the site of infection. Things like tetanus and diphtheria. These toxins get into the blood, that blood circulates through the body, dumps it into other tissues where you begin to have the disease process taking place on another part of the body. Intoxications are going to be diseases where the toxin is going to be ingested. Diseases like botulism. Botulism is a very scary disease, but we can also look at things like food poisoning. We have consumed food that is not good anymore. It's gone bad, it's gone rancid, and we've eaten it, and we end up with what is called food intoxication. And so you can have these intoxications that can be very mild, or you can have them that are going to be very dangerous, like botulinum. You have some other schemes of classification of toxins, and we can classify these as endotoxins or exotoxins. Exotoxins are going to be secreted by bacterial cells that are alive, and they are going to infect tissues. They have a very specific target, and they are extremely powerful. They are not going to be something that's just going to kind of flit here and there. These take a very small amount of protein, and they are going to bind to a very specific target tissue and begin to cause death of that tissue. Endotoxins are going to be shed from the outer membrane of gram-negative cells. Generally, when the cell dies, it's going to lyse, and it's going to release that lipopolysaccharide of the gram-negative cell wall, and it's going to have more of a systemic effect, not so targeted like the exotoxin, but it has a very systemic effect, and the signs and symptoms then are going to be very systemic. This is a great table. It tells you the differences between endo and exotoxins. Please take some time to look over this, because this is important material. I want you to be sure to understand exotoxins and endotoxins and how they can be related. Let's look at the process of infection. Now we've got these bacteria that are inside the body, and we need to then see what happens once they get in there and how we go about taking care of them. I'm going to talk to you a bit about the whole process of infection and the stages of infection. And the material that I'm talking about here is on page 376 in your book. It's called Insight 13.3, The Classical Stages of Clinical Infection. So I don't want you skipping this because we don't generally talk about those. This, I think, is very important information. So please take some time to study that graph and to read through this classical stages. The establishment, spread, and pathological effects of bacteria are going to settle in a particular target cell. You're going to have these microbes getting in, establishing, and spreading in there. They're going to cause some sort of damage that is going to be a target in that's going to target a specific tissue or organ. And once they are in there, they're going to continue to cause damage at that state. And as they continue to cause this damage, this is going to account for the stages of infection. There are 4 stages of infection. The first one is incubation, and this is the time from initial contact to the onset of first symptoms. This is the time where we're going to begin to multiply the number of bacteria until we get to that infecting dose. Once we reach that, now we're going to start seeing symptoms. The next stage is the prodromal phase. This is where you have notable symptoms. You have a very vague feeling of discomfort. You don't really feel well, but you're not at that critical stage where symptoms are going to be at their highest. Then you've got the period of invasion. This is where the agent multiplies and is going to exhibit very high toxicity, very high pathogenicity, and it's going to become established in the tissues. This is going to be the height of infection. This is where you are going to feel the very worst. This is generally going to be the longest portion of the stages of infection. And then you have the convalescent period. This is the period of recovery. This is where the patient is going to begin to get their strength back. They're going to begin to get their general health back, and they're going to start feeling better. They're going to generally return to good health. This phase can be long or it can be short, depending on the pathogen that has caused the disease. You can have this accumulated damage that's going to lead to cell and tissue death, and this cell and tissue death is called necrosis. So when you have necrosis to these tissues, this is going to lead to greater damage and can often need some sort of medical intervention. You also have some patterns of infection. You have the stages of infection, and now we want to look at how those infections can be put into specific patterns. The first thing that we want to look at is a localized infection. If you will look on page 377 on figure 13.7 and then 13.8, these are some very important figures that will help you understand this process. The first one is going to be the occurrence of infection, and we're going to look at the different patterns. And then the second one is going to be the steps involved in causing a disease in a host. And I want you to be sure that you understand how you find that portal of entry, how and what those are, how they attach, how they survive immune responses. We've talked about all of these. And then the damage. And we're going to start talking about direct versus indirect damage here in a minute. So if we look at localized infections, these infections are going to be microbes that enter the body, and they're going to remain confined to specific tissues. So you get a sore throat, and the only part of your body that begins to show any kind of symptomology, you begin to have some sort of disease process, but it stays right there. That is a localized infection. A systemic infection is infection that spreads to several sites or tissues, fluids, usually via the bloodstream. You've got bacteria that have gotten into the bloodstream, and now it is system-wide, system by system, and you're going to be you're going to have infection in a number of different places. A focal infection is an infection that is caused when you have an agent that has initially been a local infection, and you've had an agent that breaks loose from that and begins to infect other tissues. It gets carried by some process into other tissues, and there it's going to start to infect other tissues. A mixed infection is where you have several agents that establish an infection simultaneously. They're generally going to be at the same infection site, and you have a number of different microbes that are going to contribute to the disease process. And then you have primary and secondary infection. This is going to be infection according to sequence. A primary infection is going to be complicated by another secondary infection from a different microbe. So you have a primary infection, say of the urinary tract infection, and those organisms then are going to be complicated. That whole disease process is going to be complicated by a secondary organism, say from the vaginal tract, that now gets into the bladder, and you end up now with a complication from this second organism. If you look here at this picture, this is the picture that is 13.7, and you can see that you have all of these different diagrams of the localized infection, the systemic versus the focal, the mixed, and then the primary, secondary chronological infection. When we have an infection, we're going to see some sort of pathological change in tissues. And when we see those pathological changes, they're going to be accompanied by signs and symptoms. Let's be sure we understand the difference between a sign, a symptom, and a syndrome. A sign is any observable objective evidence of a disease. You can see it, okay? Chicken pox, you see the blisters, the pustules that come out on the skin, that is a sign. A symptom is going to be subjective, okay? This is going to be a subjective measure or subjective evidence that a disease has by a patient. This is going to be something that we feel, like a fever or a headache. That's going to be a symptom. And then a syndrome is when you have a disease that can be identified or defined by a complex of signs and symptoms. You have these visible signs and these sensed symptoms, and when you have all of those together, you have a syndrome. If you look here on this is table 13.8. It's on page 378 in your textbook. You can see what are different signs and then symptoms, okay? A fever is a sign. We can observe a fever. We can put a thermometer in someone's mouth. We can check that temperature, and we see that we have an elevated temperature. So that's a sign. We can have chills, meaning that we feel cold, or we have a headache, or we have fatigue. These are all going to be symptoms. These are not things that we can measure. I can't measure your fatigue, okay? I can't measure your headache or your sore throat. So these are all things that are symptoms. So signs are observable, symptoms are sensed by the body. Sometimes you can have an infection that does go unnoticed. A lot of times you have infections that produce no symptoms at all, even though you have very active microbial activity. You have microbial infection, but there are no symptoms. And these infections are known as asymptomatic or subclinical infections. Most of these infections do have some, or most infections do have some sort of a sign or some sort of symptom, but there are some that do go completely unnoticed. And these are ones that can cause some significant public health risks. One of the things that we'll probably talk about a bit when we get into the sexually transmitted disease is that a lot of women are asymptomatic that have gonorrhea. These women can then go and have sex with multiple partners, not knowing that they're passing gonorrhea on to their partners. They have no symptoms, so they have no reason to get checked, so they don't know that they're passing these on. So they can cause a public health risk. But if you don't know, and if you don't have any symptoms, there really isn't any way to determine whether or not you're sick. When we have microbes getting into the body and they've caused disease, we also need to have ways for them to get out of the body, or portals of exit. These are ways that pathogens are going to vacate the human body. They're going to depart through specific portals of entry, just like they enter through specific portals of entry. In most cases, a pathogen is either going to be shed or released from the body through either some sort of secretion, some sort of excretion, some sort of discharge, or sloughing of tissues. You have, just like you have specific entries, portals of entry, we have respiratory and salivary portals of exit. The mucus, the sputum, nasal discharge, and any other moist secretion that escapes from the body is going to be a media that is effective at moving these microbes out of the body. The most effective means of portals of exit are going to be coughing and sneezing, and then talking and laughing. These are ways that we produce a lot of air that leaves our body and takes a lot of the microbes with it. When we talk about portals of exit, these are going to be ways that we get rid of bacteria, not ways that bacteria are finding a way to get out. They want to stay in. We want them out. So these portals of exit are going to be to our benefit. Skin scales, these are going to be portals of exit where we have the skin and the scalp constantly being sloughed off. That outer layer of skin is constantly being removed. We are going to slough off billions of skin cells in a single day. When you have skin that's not intact, that may not be healthy, you have lesions, or you have lesions that are oozing pus or some sort of exudate, those can also be portals of exit because they're trying to remove any of the bacteria that's in there and trying to get rid of it. The fecal portals of exit are probably a very common portal of exit. We all poop. I think they've written a book about that one. Everybody poops. These intestinal pathogens can cause that should be which and not which, can cause an inflammatory response that's going to cause gastric motility, which is going to lead to the increased release of that pathogen. When you have diarrhea, you need to try not to medicate it, because that's your body's way of saying, I need to get this out, and that feces then is going to leave, and the microbes that are connected with it. Now, obviously, you need to keep yourself from being dehydrated, but if you can take things that don't stop the diarrhea response, like Imodium, things like that, and just kind of stay home and let it run its course, that is the best thing that you can do. A lot of times feces contains a lot of pathogens of public health significance. You've got things like hepatitis, you've got things like Salmonella and Shigella and E. coli, numbers of different things. These can often be a possible source of contaminated drinking water. When you've got feces that gets into the water, and that water is the source of your drinking water, you can end up then consuming it and causing a reinfection. We can also have contamination when crops are fertilized with feces, animal feces, things like cattle poop, cow dung that gets used to fertilize crops. You've got E. coli in there that is not part of our normal E. coli and can be very dangerous, and we can end up getting sick. The exit portals of the urogenital tract, basically you're going to have a number of these agents that are going to be removed through vaginal discharge, through semen release. Sometimes you can have pathogens that enter the kidney and are released in the urine. This is going to be less common. Most of the time when you have pathogens released in the urine, these are coming from the bladder. If you've got a bladder infection that's worked its way up to the kidneys, you're generally going to see blood in the urine. You're going to have a high white cell count, and you're going to have, those are the times where you're going to start to see pathogens released in the urine from the kidneys. Most of the time they're going to come from the bladder. We can also have portals of exit from bleeding or from bloodletting when we act as some sort of portal through some sort of vascular puncture, whether it's a natural puncture or an artificial puncture. When we pull blood from the body, either through a hypodermic or through a wound, we're going to be releasing microorganisms that may be in the blood. Blood feeding animals like ticks and fleas are common transmitters, and so we can have blood taken from one organism and transmitted to another organism by some of these blood feeding animals. And we're going to talk about what these are here in a few minutes. Sometimes when you have a person who has been sick and they recover, but they don't necessarily remove the microbe completely, you have these patients that have become what are called latent carriers. When you have latency, this is the dormant state of a microorganism. The microorganism, once it's in that dormant state, is going to stay quiet. And all of us, I'm sure, carry dormant organisms throughout our body. But sometimes these can periodically become active and they can cause a reoccurrence of the disease. People with latent infections may or may not shed the agent in any sort of one of these portals of exit. But those who do shed their organisms are what we call chronic carriers can act as a risk of infection for the rest of the population. So knowing what you are releasing from your portals of exit can be relatively important depending on the disease. If you are somebody who has had hepatitis or who has had typhoid, something like that, your feces is going to be very important, and what you release in your feces is of clinical significance. An example of some of these organisms that become latent, the disease that causes mononucleosis, the Epstein-Barr virus, can persist in a host for months, even years, and you can have relapsing and recurring episodes of mononucleosis. Some diseases can leave what we call a sequela, which is basically a long-term or potentially permanent damage to the tissues or organs. You may not have a complete function of the organ or the tissue for a long period of time or ever at all, and this is basically called a generalized sequela. Disease transfer is going to require some specific pathogens, and some of those pathogens are going to live in places other than the human body. They're going to live in some place called a reservoir, and that pathogen has to have some sort of permanent place to reside so that it can persist or continue to exist in the environment and spread to other places so that it can divide and replicate. The primary habitat for a pathogen in the natural world is called its reservoir. Okay, this is where it lives when it is not causing disease. You have an individual or an object where the infection is actually acquired. This is termed the source. So a reservoir and a source are two different things. A reservoir is where the pathogen lives all the time. It's its natural habitat. It's where it lives. It's where it originates, and this is the reservoir. Where the infection is acquired is the source. So be sure you understand the difference between reservoir and source. We have living versus non-living reservoirs, and a living reservoir is an individual who inconspicuously harbors a pathogen. He doesn't realize that he harbors a pathogen and spreads it to others without knowing it. These are people that are called carriers. So a carrier is a living reservoir, and we have a number of different carriers. If you look on page 380 in your book at figure 1310, you'll see that you have a number of different carrier states. You have this asymptomatic carrier. This is someone that shows no symptoms and has no idea that they even have an infection. Then you have the incubation carrier. This is a carrier where they spread the agent during the incubation period of that clinical infection. And then you have convalescent carriers. These are recuperating patients that have no symptoms but continue to carry microbes, and they shed them either in their mucus or their feces or their urine, and they can then pass it on to another individual. Chronic carriers, these are going to harbor the agent for a long period of time after recovery. Sometimes you can have lifelong chronic carriers. These are people who have carried the organism for the rest of their lives after they've been infected and have gotten better. And then you have a passive carrier. This is somebody who accidentally transfers it from person to person. A good example of this is a nurse from one patient to another. You have these nurses that go and they handle one patient, and then they possibly don't get their hands washed completely, and they then pass that microbe on to another patient. If you'll remind me, I will tell you about my experience with when I had my thyroid taken out. Here's the picture 13.9 that is an example of all of these different carriers, the asymptomatic versus the incubation, convalescent, chronic, and then the passive carrier. You can have animals act as reservoir sources too. A live animal can transmit an infection from one host to another, and when that animal does this, then it is called a vector. And these animals that are going to transmit these infections can transmit them from host to host. These can be either biological vectors or mechanical vectors. And I'm sorry for all the typos that I'm seeing here. These animals are going to act as vehicles for transmission. If you think about a vector as a vehicle, it moves this organism from one place to another. They don't cause the disease, but they take that agent from one place to another, from its reservoir to its host, from one host to another. So you've got a way to move this organism from place to place. A majority of these vectors are going to be arthropods, things like mosquitoes and flies and fleas and ticks. These are going to be the kinds of vectors that we have. You can have large animals also be vectors. When you're talking about a rabid animal, a mammal that has been infected with rabies, it can also be a vector, or that may carry rabies but is not showing symptoms. It would be a vector. Again, vectors can be divided into biological vectors, which are going to be actively participating in the pathogen's life cycle, meaning that at some point this pathogen is going to incubate within the life cycle of this arthropod. You're going to have all of the stages within the pathogen's life cycle taking place in this organism. Things like mosquitoes and fleas and ticks. These are going to be biological vectors. Then you have mechanical vectors. These are going to be vectors that transport an infectious agent without being infected. You've got a fly that lands on feces that has Salmonella typhi in it. Now this fly has feces on its feet, and it goes then and lands on your food, and then now has contaminated your food, and you consume that food, and now you've become sick. So that fly now is a mechanical vector. These biological vectors can transmit agents to a host through either biting, through an aerosol formation, or by touch. And if you look at this picture here, you'll see that you've got a mosquito here as a biological vector, and then you've got a housefly, a common housefly here as a mechanical vector. When you have these biting vectors, they can either inject infected saliva into the blood like a mosquito does. It can defecate around a wound or a bite. If you've got a flea that has bitten you and then you have that flea defecates around that wound, it can get in through the skin, and that vector now has passed that disease on to you. Or it can regurgitate blood into a bite, into that wound, like the tsetse fly. The tsetse fly is a fly in Africa, very common a fly in Africa that can cause significant damage. The flea can also regurgitate into a body site. The common flea is the vector for the disease that causes plague. Zoonosis is, we can have animals that can naturally transmit disease to humans. These infections are called zoonosis. This is an infection from an animal to a human by some natural process. Humans do not contribute to the persistence of this microbe. They don't continue to give it back. Generally, the human is the dead end host. It can have multi-host involvement. You can have a number of different animals that are part of this zoonotic disease process. There are at least 150 worldwide zoonotic diseases that we know of right now. Here are some of the zoonotic diseases. You can see viruses versus bacteria versus some miscellaneous worms or fungi, things like that. And you can see what their primary animal reservoir is and how we can end up with these organisms from some sort of interaction with these animals. And they don't necessarily even have to come by a bite. They can be from some aerosolized mucus, or we can consume them, and we can end up getting sick then from this animal. Just like we have living reservoirs, we've got non-living reservoirs. We have contact with environmental sources that don't act as some sort of living place for these organisms to live. These are going to include things like soil and water. Soil harbors a number of vegetative organisms, and these are nearly coming from all microbial categories, whether they're bacteria, viruses, fungi, parasitic worms. These all are going to live in the soil. You're going to have examples of all of these in the soil. Water has very few nutrients, but it can still support a number of pathogenic species. Things like Legionella or Cryptosporidium or Giardia. These are all different pathogens that live in the water and can subsist in the water and can make you sick. A lot of times in the summertime, if you have a pool that is not completely chlorinated or doesn't have enough salt in it to keep it clean, we can end up with an outbreak of Cryptosporidium or cryptosporidiosis, and it's because this Cryptosporidium, this fungi, is living in the water and subsisting in that water. We have our vectors, we have our reservoirs, and they all are going to contain our organisms. And now we need to figure out how we acquire and how we transmit these infectious agents from place to place. An infectious disease can be categorized by how it's acquired. How do we get it? Do we get it from an animal? Do we get it from another person? Do we have to have direct contact? Can we get it indirectly? So when the infected host can transmit an infectious agent to another person and establish an infection, that host has a communicable disease. This is going to be a person-to-person contact. Okay? You have a communicable disease. I can cough on you. You can take that organism in. Now you're going to get sick. Transmission can either be direct or indirect. When we have some of these agents that can get passed from person to person, we say that these are contagious agents, meaning that they are highly communicable. These are going to be things that are very easily spread from one host to another. When that infection does not arise through the transmission of some sort of infectious agent from one host to another, otherwise it's gotten from the environment or we've consumed it, it's come through some special circumstance, this is called a non-communicable disease. A good example of this would be food poisoning. If I get food poisoning and I throw up on your shoes, you are not necessarily going to get sick from the stuff that's now on your shoes. There aren't any microbes in there that you're going to get that are going to cause you to be sick. These are going to be acquired through some special circumstance, like food poisoning. Compromised people that have a compromised immune system can be infected by their own microbiota, but because it is their own, again, this is a non-communicable disease. You can have an individual that does get some sort of accidental microbe from some non-living reservoir like the soil or the water, like when I was talking about the cryptosporidiosis from swimming pools. These again are going to be non-communicable diseases. These, we said, can these diseases can be spread either directly or indirectly, and this contact can either be what we call horizontal, where it's spread from one individual to another in some sort of horizontal mechanism, or vertical, where we have transmission from patient to offspring, and this is going to occur through the ovum, through the sperm, through the placenta, or through the breast milk. This is going to be a vertical transmission. When you're talking about a baby that hasn't been born yet, and that baby ends up then having some microbe cross the placenta, that's going to be a vertical transmission. Contact transmission means that you're going to have some type of contact that has to occur between the skin or mucous membranes of an infected individual. Things like kissing, things like aerosolized droplets from a sneeze or a cough, or somebody that has chicken pox and breathes that out in their, just in their breath. If you sit there and blow on your hand, you're going to feel moisture on your hand. So we aerosolize a lot of our microbes come out just in the process of talking. Direct contact is going to be those vertical transmissions. Obviously, breastfeeding is going to be a vertical transmission. This is going to be coming in contact. In utero infections, vertical transmission, and then some sort of biological vector. If you have some sort of direct contact with a biological vector like a mosquito or a flea, then those are going to be direct contact transmission. Indirect transmission, that means that this agent has to be passed from an infected host to an intermediate conveyor and then to another host, meaning that I'm going to get sick, I'm going to cough on my hand, I'm going to put my hand on my phone receiver, and I've deposited those bacteria and those viruses now in the receiver of my phone. If somebody then comes and picks up my phone and uses the receiver, it now has those microorganisms, that person now has those microorganisms on their hand, and they can then transmit that then to their own nose or their mouth or something like that. When you have these intermediate conveyors, these are going to be inanimate objects. These are going to be things that are used commonly and can transmit infectious agents and act as a vehicle for transmission. These inanimate things are going to be things like food, like water, biological products like medications or things like that, and then things called fomites. A fomite is an inanimate object that can harbor organisms and transmit those pathogens to another person, like the receiver of my telephone. That is a fomite. You can also have air as a vehicle for transmission. Indoor air especially can have a number of different organisms in it. It can act as a medium for suspension and dispersal of a number of respiratory pathogens via the aerosolization of our mucus as it comes out of our mouth, and it will hang then in the air. Or we have something called a droplet nuclei. This droplet nuclei is essentially the dried residue from mucus and saliva that gets ejected from the nose and mouth. It can sit on an inanimate surface or hang in the air for a very long time, and then we can breathe it in. This can be encountered by a new host. This droplet nuclei can be encountered by a new host either geographically or chronologically distant from the original person to release that droplet out into the environment. If I cough in the hallway, that droplet nuclei can make its way down into the lobby of the 1500 building, and somebody can breathe it in. That's a significant distance from my office if I'm standing outside my office in the hallway. So you can see how these can cause a significant amount of illness if you have somebody that has an unstifled sneeze or an unprotected cough or something like that. These droplet nuclei are considered indirect contact. These are not like the droplets that you have through the direct contact where you're breathing them out of one person and breathing it into the next. Aerosols basically are going to be suspensions of a very fine dust or very fine moisture particles in the air that are going to contain live pathogens. These are not going to be those dried out residues, but these are going to be moist particles that still contain these organisms. Things like urine, that's a good example of an aerosolized suspension. When urine dries and you sweep it up, it can give you those droplet nuclei, or that urine can and feces can aerosolize, and you can end up then with those aerosols in the environment. Here's a picture of a communicable disease. Remember, communicable is host to host. You're going to have direct versus your indirect vehicles. And so this is a pretty self-explanatory, I think, figure. So take some time to look over it. Make sure you understand all of the processes that can happen, and you can end up then knowing which one is direct, which one is indirect. Understand that they are all communicable. Here is a sneeze. I used to have this picture on my office door until I had to take everything off my office door down. But if you look, think about when you sneeze. This is essentially what your sneeze looks like, and every single one of those droplets harbors thousands, if not millions, or tens or hundreds of thousands of microorganisms per drop. And you end up with those organisms subsisting in the air and being consumed or being taken in by other people. That is one hefty sneeze. We can have infections that we get in places where we would certainly not expect to get an infection, and that's the hospital. Nosocomial infections are infectious diseases that we get when we are in the hospital. We either acquire them or they develop during a hospital stay. Patients can become infected during a surgical procedure, or when their lowered immunity allows some sort of normal flora to go and invade body tissues where it doesn't normally invade, and you end up then with some sort of infection. Other patients can acquire infections from fomites within the hospital, medical equipment, other patients, hospital personnel, as well as visitors. Visitors are probably one of the #1 ways that we bring infection into the hospital. The air and water in hospitals, if you're not in a particular area in the hospital where the air is sanitized or filtered, you're going to get the air from the room next door and in the hallway and everything else. There are between 2 and 4 million cases of nosocomial infections a year. This leads to about $5 to $10 billion of increased hospitalization costs every year. So it's important to carry out medical asepsis, to understand those asepsis procedures to keep from passing these infections to patients, and from patient to patient or patient to worker and back. The most common infection are going to be urinary tract infections, respiratory tract infections, surgical incisions, as well as gram-negative intestinal biota. These are all going to be areas where you have significant exposure to bacteria. This is a good graph to show you where some of these nosocomial infections are coming from. Again, most of them, 40% of them, are urinary tract infections, and that generally is because most of the time when you're in the hospital you have some sort of indwelling catheter. You get a Foley catheter or something like that, and a lot of times you end up with urinary tract infections. Surgical site infections and respiratory tract infections are pretty close. Surgical site infections, just simply because bacteria are everywhere, and unless you have some precautions in place, a lot of times those surgical sites are going to get infected. Respiratory infections, breathing in what other people are bringing in, and having a lower immune response, is going to cause you some trouble. Meningitis and gastroenteritis. These are two, they kind of contribute to this 12% of what they call other diseases. Meningitis and gastroenteritis, you wouldn't think they would be very related, but they can be. You can have gastroenteritis that becomes septic, gets into the meningeal fluids, and you end up then with gram-negative meningitis, which is never a good thing. Then skin and septicemia infections. Those two you don't see a whole lot, but you do see some increased numbers of these types of infections. Because of nosocomial infections and trying to limit the number of infections, there have been some universal blood and body fluid precautions that have been set out. Medical and dental offices are required to keep these very stringent measures to prevent these nosocomial infections. These universal precautions are guidelines that are set out by the Centers for Disease Control and Prevention, and they're used for handling bodily fluids, bodily substances. These precautions are based on the fact that you're going to assume that all patient specimens are going to harbor some sort of infectious disease. Most of us are not as clean as we'd like to think that we are, and so when you have an infection, you need to assume that it has spread everywhere. It includes body substance isolation techniques so that you can these can be used in the case of a known infection. If you know somebody that has HIV and you're taking their blood, you're going to use these BSI techniques to ensure that you don't poke yourself or that you don't infect yourself. And so these are very important universal precautions. On page 386 in your book, you have the 6 different major universal precautions that are recommended for healthcare settings. Be sure that you take some time to look at that. The last thing that I want to talk about here is epidemiology, and this is the study of disease in populations. We've looked at how microbes enter the body, how they establish themselves, and how they cause disease. And we've also now looked at the way that we can go through and transmit disease, what the reservoirs are, what the vehicles are, what the vectors that transmit these diseases. But now I want to talk about how we study the diseases in the population, and how do we know what the statistics are and what those statistics mean. When we're studying the frequency and distribution of disease and other health-related factors in populations, this is the science of epidemiology, or the study of epidemiology. The techniques of the epidemiologists are going to track health-related behaviors, things like smoking and exercise. They're also going to collect clues on agents that are causing disease. What are the pathology of these agents? Where are they coming from? What are their sources, and how do they transmit from person to person? Are they communicable or are they non-communicable? What are their numbers, and what's the distribution within the community? Are they distributing equally? Is it in one specific area in the community? How many do we have in that community? Is it a high number? Is it a low number? These are all very important statistics that an epidemiologist would look at. They're also going to be concerned with the virulence and portals of entry and exit of these specific agents. And they also want to look at the course of the disease. Is it something that takes a long time to progress, or is it an acute disease? Does it happen quickly? They're also interested in surveillance. They're going to collect, they're going to analyze, and they're going to calculate and put all of this data, and they're going to look at rates of occurrence. How fast is it occurring? Do we have 2 or 3 cases a year? Do the cases increase or decrease at certain times during the year? What is the mortality? How many people die from this? What is the morbidity? How many cases of this are there? So we're going to look at morbidity and mortality, and see, is there a correlation between the two? Do I have a high morbidity with lots of cases but very few people die? Or do I have a low morbidity with very few cases but the mortality rate is 50% or higher? So those are going to be the things that an epidemiologist is going to look at. One of the other things that they're very interested in are these things called reportable diseases. And these are diseases that by law must be reported to authorities like the public health authorities. And these are then going to be sent to the Centers for Disease Control, where they will be compiled by the epidemiologists and put out in a weekly notice called the Morbidity and Mortality Report. This is a very interesting, I used to get this all the time, and I unsubscribed to it because I was getting more and more than I could read. But your textbook on page 388 gives you the website www.cdc.gov/mmwr, and this gives you the morbidity mortality report, and it's free, and it's a very interesting thing to read. You can see the progression of diseases, specifically when you're looking at like during the flu season. Or there's been an outbreak somewhere in the world from some disease that is not very common, and you can read the morbidity and mortality weekly report, and it's very interesting reading if you ever want to log on, because you can log on and read it online. Once the CDC has this information, they're going to share this information with the World Health Organization. And these are going to be people that look at epidemiological trends worldwide. They're not going to just look at the United States. They're not going to just look at, like North America, South America, Africa. They're going to look at this worldwide. And they can then do a lot of preventative work once they know these statistics. Just like everything else we've been talking about, there's some terminology that we want to be sure we understand. Prevalence is basically the total number of existing cases with respect to the entire population. In other words, how many cases do I have divided by the total number of people in our population. So if we look at our class, say our class has 21 people, and in that we have let's say we have 20 people, easier math. Let's say we have 20 people, and we have 4 of you that have some disease. So we would take the 4 of you divided by our population of 20 people and multiply that by 100, and we would then have the prevalence, which would be a 20% prevalence of a particular disease in our population. Incidence is the number of new cases over a certain time period. So we're going to look at the number of new cases divided by the total number of susceptible people. So we have to know, when we're talking about looking at incidence, we would have to know how many people in the population are susceptible to a particular disease. If we're talking about chickenpox, we'd have to know how many people have had chickenpox in our population, how many people have been vaccinated in our population, and then we would see how many are left over that would be susceptible. And then we would look at the new cases, divide it by the number of susceptible people, and we would have our incidence. Mortality rate is the number of deaths in a population due to a particular disease, and morbidity is the number of people that have gotten sick with this particular infectious disease. If we talk about food poisoning is one of those reportable diseases, and so the morbidity rate may be very high, but the mortality rate is relatively low. Not very many people die from food poisoning. But if we look at botulism intoxication, the morbidity is relatively low. You don't see very many people being afflicted with botulism, but the mortality rate is in excess of 50%. So it has very high mortality, even though it has very low morbidity. Here are some graphs that you can look at when you talk about the different epidemiological data. If we look at graph A, this is basically a time frame. It's basically the incidence of hepatitis per year in the United States between 1966 and 2006. And you'll see that the different types of hepatitis are on the decline. And you'll look here at 1966, about 1970, we had very acute hepatitis. This is no accident. This is about the time you begin to see a lot of IV drug use. A lot of people in the hippie culture when IV drug use was very prevalent and people were living in how do I want to say this, in collections or groups of people, and they would share everything. And so you had this very high peak of acute hepatitis A. But as you can see, it's declined. We did have another peak in between 1985 and 1987. But now if you look at 2006, it's declined to a number or fewer than 200,000 cases in the population at any given time. If you look at hepatitis B, we had a high peak there around 1985, and that was kind of at the beginning of when HIV was identified. You had people that had a combined infection with acute hepatitis B and HIV at the same time. And then you'll notice that we didn't start looking at hepatitis C until 1990, and this kind of coincides with a lot of the immigration of people. They called them the boat people, the people that were coming across from Cuba, coming across from some of these other African countries where hepatitis is more prevalent, hepatitis C. But we didn't start taking any data on that until 1990 because hepatitis C was not, you didn't have very many cases, if any at all, earlier than that. If you look at the graph B, this is basically looking at HPV infections among young adults between the years of 20 and 29 years. It can also divide things by ethnicity, by education, percent poverty, as well as sexual partners. So you can get a lot of information from this graph here. And then the bottom graph here is basically the malaria activity in 2009. And you can see where in the world malaria is endemic, where in the world malaria has a presence, and then where in the world you have no malaria at all. And you can see a good deal of the world doesn't have a lot of malaria. But if you look here in parts of South America, parts of Africa, parts of Asia, you see that malaria is endemic. Epidemiological frequency is important. We need to know what is the frequency of particular cases. When you begin to have increases in disease in a specific geographical area, it's good to know what is the source of that infection, or what is the source of that epidemic. Is it a point source epidemic? Is it a common source epidemic? Or is it a propagated source epidemic? Point source epidemics are epidemics in which an infectious agent came from a single source. All of the patients are exposed from the same source, particularly a foodborne illness. You have a big, huge, if you think about, well, so many of you are too young to remember this, but Jack in the Box. Jack in the Box had its meat come from one specific place, and this meat then was consumed, and you had a number of people with these foodborne illnesses. Some of them died, but it was all from the same source. Common source epidemics are outbreaks that result from a common exposure of a single source over a period of time. Say you have contaminated drinking water. You have a number of patients that get the same disease over a period of time. They use the same water source, the same drinking water source, but they didn't all drink from it at the same time. They drank from it over a period of time. And so you had a number of different people. The number of cases is going to fluctuate over time. And then a propagated epidemic is when you have an agent that is a communicable disease and therefore can be propagated over time. It goes from host to host to host to host. It's not necessarily a common or a point source. It goes wherever it can go. And when you look at a propagated graph, you're going to start very low and it's going to get very high, and then it will eventually start to taper off as recovery begins to take place. And if you look at the graph here to the right, you're going to see that we started out with a very low number of cases, and as that began to sustain itself in the environment, that agent began to sustain itself and propagated from host to host, you'll see we have a very high increased number of organisms. There are some other terms that are going to help describe cases and patterns of epidemics. One of the terms is index case. This is the first patient found in an epidemiological investigation, or the patient that was brought to the attention of the epidemiologist. This doesn't necessarily mean it's the first patient to have the disease, but it is the first patient that is recognized as having a disease. A lot of times these will be named patient 0. Okay? Endemic basically is a disease where the infectious disease is going to be at a steady frequency over a long period of time. You have these diseases that are going to be in places all the time. We've looked at the map on malaria. You'll see that in a good part of Central Africa, you're going to have malaria there all the time. It is everywhere. And you have them at a very steady frequency. The numbers don't change. They don't increase and they really don't decrease. And they do this over a long period of time in a particular geographic locale. Sporadic disease are going to be occasional cases that are reported at irregular intervals in random locales. They're going to pop up here and pop up there, and there's no real interval between. If we look at influenza, we see that it comes around November, December, and stays until February, March. And we have that interval. We recognize that, and that's going to be an epidemic that we can recognize. But sporadic are going to be occasional cases here and there at irregular intervals at random places. Epidemic is going to be the prevalence of a disease that increases beyond the expected numbers for that population. When we have the influenza epidemic, we end up then having a number of cases where we have let's go back. Let's look at, in the book, it talks about food poisoning. And so you end up then with a number of cases that is far beyond the expected number in the population. And so that is what we call an epidemic. And then a pandemic, this is where you have the spread of an epidemic across continents, where we're starting to jump borders, we're jumping oceans. And so you're going to have this pandemic. The 1918 influenza pandemic circumnavigated the globe in 6 months. If you sit there and think about the fact that there wasn't air travel like we have today, that pandemic went very rapidly. We can have a pandemic in the United States, or excuse me, a pandemic in the world right now within 24 hours. We can jump on a plane, we can be sick, jump on a plane, and end up someplace else in the world, begin to spread this continent to continent, and we would end up with a pandemic very rapidly. Here's a map. You shows some endemic occurrences. You've got them all down in this locale. They're always persistent, and they're just always here at a particular place. Epidemics, you're going to have a significant number of patients, people that have the disease that are higher numbers than the population would be expected to have. So this would be an epidemic. This is going to be sporadic. You've got diseases popping up just kind of everywhere with no specific interval, no specific relationship between the different sites of infection. And then here obviously is your pandemic, going from place to place to place, and you are ending up then jumping borders. It may start in one place and then jump oceans, jump borders, and move into other areas of the world. Okay, this is the end of chapter 13. I have the Learn Smart up on Connect. It's ready to go for you for chapter 13. There's also a quiz for chapter 13, so please be sure you take some time to look at that, and take time to look at due dates. Please, please, please. I'm getting to the end of the semester. I am less likely to extend these due dates because I need to be able to keep up on my grading. So take time to look at the due dates, make sure that you are observing those and not needing to come to me and say, hey, can you extend the date? Because I'm more than likely going to tell you, no, I can't. So happy listening, and I'll see you in class.